Welcome to SEQNEXT-HLA, the software solution for efficient and reliable HLA analysis of your next-generation sequencing data. In this video, we will provide an overview of the software, the analysis workflow, and key features for the evaluation and validation of HLA next-generation sequencing data. JSI Medical Systems is a German company headquartered in the Black Forest and has been active in the field of bioinformatics software solutions since 1999. With more than 25 years of experience in the analysis and interpretation of sequence data, we develop software solutions in close collaboration with university and private genetic institutes. Our customers receive personal support from our highly qualified expert team for all software-related questions, including the integration into existing laboratory workflows. Today, our products are used worldwide in more than 400 installations. The product portfolio of JSI Medical Systems covers a broad spectrum of molecular genetic applications. This includes solutions for next-generation sequencing, Sanger sequencing, microarray analyses, MLPA, as well as import and export interfaces. Sequence Pilot is a CE-IVD certified software, and SEQNEXT-HLA is our specialized solution for HLA typing using next-generation sequencing. The software supports a clearly defined analysis workflow, ranging from raw data processing to final validation. The raw data import into SEQNEXT-HLA is manufacturer and kit independent and supports all common data formats from modern NGS sequencing platforms. The analysis is performed automatically based on an integrated and regularly updated HLA database. Data analysis in SEQNEXT-HLA is characterized by high sensitivity and specificity. Not only common alleles, but also rare and novel alleles are reliably detected. In addition, the software supports the detection of pseudogenes, thereby contributing to a correct interpretation of the results. The analysis is highly efficient and usually requires only about 1 minute. Analysis settings and warning messages can be customized individually, allowing the workflow to be optimally adapted to the requirements of each laboratory. In addition, SEQNEXT-HLA provides detailed analysis information. Furthermore, unusual results are not finalized automatically, but are specifically flagged. This enables controlled manual validation, while uncritical cases can be processed fully automatically. This combination of automation and targeted manual verification forms the core concept of SEQNEXT-HLA and supports an efficient, transparent, and validated analysis process. The validated results can then be exported or automatically transferred to a laboratory information system. After this short overview, we will now switch directly to the software and take a look at the workflow using concrete examples. The validated target regions, as well as the corresponding analysis settings for the ProTrans kits, can be conveniently imported in a single step. In the next step, the raw data can be loaded directly into the software. The import can be performed either automatically or manually, depending on the requirements of your laboratory. After the import, the data is merged in the joining section. This is where the validation of the analysis results takes place, presented clearly and transparently, and supported by the predefined quality parameters. In the result overview, you receive a complete overview of the analyzed order. Displayed are the read DNA number, all analyzed HLA genes, as well as the corresponding typing results. In addition, a clear coverage visualization is available, allowing a quick assessment of the data quality. The result presentation in SEQNEXT-HLA is flexibly configurable. The resolution, including G or P groups, can be defined either at the order level or at the gene level. These settings can also be defined individually for report generation, as well as for the transfer to a laboratory information system. In addition, a central overview is available in which all possible resolutions of an order are transparently displayed. This allows different levels of resolution to be quickly compared and also exported. The evaluation score supports you in assessing the analysis results. At a glance, it indicates whether a result is uncritical or should be examined in more detail. First, we look at gene A with a high evaluation score and a heterozygous result. In the sequence window, a detailed representation of the data is available. The exon and intron structure is displayed clearly. Heterozygous positions are highlighted in blue below this view. Coverage is displayed separately for haplotype 1 and haplotype 2 and allows a targeted quality assessment. Below this, the database sequence, the combined sequence of both haplotypes, as well as the individual haplotype sequences with their assigned reads are displayed. The allele combination calculated by the software is shown directly in the results section and as the first entry in the table below, corresponding to the result displayed in the joining overview. This result can be manually overwritten, or alternatively, a different, less likely combination can be selected from the table. If an allele combination with a mismatch is selected, the corresponding position is marked in red in the exon-intron overview. In the next step, we look at another gene from the same sample that shows a low evaluation score. Point deductions in the evaluation score occur, for example, in cases of limited data quality, such as insufficient coverage, the presence of background alleles, or when automatic result calculation is not possible. A typical example is the manual acceptance of a new allele. For this, we take a closer look at the HLA gene DRB1. In such cases, the gene is clearly marked with check results via the evaluation hint. This indicates that the affected gene must be reviewed in the detailed view within the sequence operation. When opening the detailed view by double-clicking on the gene, the software automatically jumps to the relevant position that led to the identification of a new allele. The deviation is located in exon 2 at position 270. Based on this sequence information, the result is labeled as a new allele with the highest similarity to DRB1, 9121. This result can be manually accepted. To do this, the option New Allele is selected via a right-click on the corresponding gene and confirmed with OK in the dialog. After manually accepting the new allele, the result is automatically transferred to the joining operation. The result column for DRB1 is now fully completed, and the order can be finally evaluated. For the DRB genes, SEQNEXT-HLA automatically performs a pseudogene check. This is based on the determined DRB1 result and verifies which additional DRB genes are expected to be present. In this example, the result DRB17 and 9 is present. Based on this combination, only the presence of DRB4 is expected. Accordingly, no warnings are displayed if DRB3 and DRB5 are not detected. In the second example, we examine a case of a second order that is also marked with the evaluation hint, check result. In this case, due to a background signal that may indicate a heterozygous result. For this DNA number, the HLA gene DQB1 was initially calculated as homozygous and therefore marked with the evaluation hint, check result. When opening the sequence view, it becomes visible that although a homozygous result was called, background signals are present across the entire region of interest. For further evaluation, the Edit Results dialog is opened. This can be accessed via a right-click on the gene, selecting Editing, and then Results. In all 4 exons, 2 alternative sequences are displayed. These sequences show coverages between 11 and 14%, and therefore do not exceed the predefined thresholds of 15. For this reason, the gene is not automatically called as heterozygous. However, in order to avoid falsely homozygous results, SEQNEXT-HLA also evaluates alleles with lower coverage. Therefore, a second result is additionally listed in the dialog as possible heterozygous. To adopt this result, the corresponding entry is selected and the dialog is confirmed with OK. After accepting the heterozygous result, the previously visible background positions are no longer present. They have now been correctly assigned to the second allele. The manually edited result is automatically transferred to the joining operation. The order can now be further processed and finally evaluated based on the corrected heterozygous assignment. After these manual editing steps, the validation is complete. The order can now be marked accordingly as technically or medically validated. Afterwards, several options for further result processing are available. The results can be printed as a report, alternatively exported as a text file, or automatically transferred to a laboratory information system. In summary, SEQNEXT-HLA provides a comprehensive and validated workflow for HLA typing using next-generation sequencing. From automated analysis to targeted manual validation steps and structured result approval, the software supports a secure and traceable evaluation of sequencing data. Already in the result overview, you receive a quick overview of data quality, coverage, analysis hints, and the calculated results. The data analysis is characterized by high sensitivity and specificity. Rare and novel alleles are reliably detected, as well as pseudogenes. The analysis is highly efficient and usually requires only about 1 minute per sample. SEQNEXT-HLA is manufacturer independent and supports the analysis of sequencing data from platforms of all major providers as well as kits from different manufacturers. In the detailed view, comprehensive information is also available, including read alignments. Through this automated and transparent workflow, SEQNEXT-HLA enables fast, flexible, and reliable HLA typing. If this video has sparked your interest in our product, we would be pleased to invite you to test SEQNEXT-HLA in your laboratory with our support. For further information, please feel free to contact us. Thank you very much for your interest in SEQNEXT-HLA.